All right, last big video before our AP exam. In this video, we're going to talk about biotechnology. And really, biotechnology or DNA technology, as I have it listed here, is just a series of techniques that are used to identify, copy, cut, modify, splice DNA together in order to do something for us, right? A real modern example of DNA technology you may have heard about in the news is the CRISPR technology, and I'll kind of touch on it a little bit when I get into recombinant DNA and genetic engineering, but that's the kind of thing we're talking about. So a lot of the lab coat genetics is biotechnology. When we talk about the term genetic engineering, we're talking about directly manipulating the DNA of an organism. Okay, and that's a common term you'll hear sort of in the news, people talking about genetic engineering, and that's what that means. There are a number of techniques that are part of biotechnology that are not genetic engineering. One of the important tools in biotechnology is that of the restriction enzyme, and we talked about these in Baby Bio and talked about the fact that you can think of these as chemical scissors that are designed to cut DNA at very specific sequences. And recall that the cut end that they create is called a sticky end. And that's because, let me just kind of draw you one here. When they cut, here's a sequence of DNA, right? And this is a palindromic sequence, so it reads, the same way frontwards and backwards, right? So here's a sequence on the DNA molecule. If you have a restriction enzyme whose recognition sequence, the sequence where it cuts, is at TTAA, an enzyme that's moving along on the top strand here would cut the sequence right after that AA. But if you had another restriction enzyme reading the other side of the DNA molecule, it wouldn't cut in the same place. You wouldn't get a blunt cut straight across the DNA molecule. Because in this direction, if an enzyme was moving in this direction, this would read AATT and not TTAA. So instead, what you get is an enzyme that cuts like this, and what it creates is this stair-step shaped cut. And the DNA, and you'll see it in a picture down the line, opens up at that point, and now what you've got are these open hydrogen bonds, right? These hydrogen bonds have been broken, and they would be opened up when the cut occurs here and here. And that's why they're called sticky, because those hydrogen bonds are available for other things to kind of stick onto them. So it's easy to splice pieces of DNA together so that they stick. All right, let's look first at how we can cut and take a look at DNA sequences. Okay, and to do this, it's really a two-step process. RFLP is what you're seeing right here, and that stands for Restriction Fragment Length Polymorphism. Okay, and again, that's RFLP. Okay, and that's what you see there. Fancy name, really it means you put some restriction enzymes and a solution of DNA together, and you just let it sit quietly in an incubator, and the DNA gets cut up by those restriction enzymes. Just let the same restriction enzymes I just mentioned, they're used to cut the DNA up. The problem is, what you see if you look at these little tiny test tubes here is really just a little tiny slime ball of DNA in the bottom of each of these. So in order to visualize the DNA fragments that we've created through RFLP, we need to use gel electrophoresis. Okay, and this involves taking a gel right here, and we put these little deep wells in the gel, these holes, and you can actually pipette samples from our DNA there into the gel, and DNA has a slight negative charge, a partial negative charge. So when we put the electricity on, the samples will run across the gel toward the positive electrode or the anode. And they will spread themselves out. The reason for that is that the gel offers some friction, so the longer molecules end up kind of getting stuck as they move along. There's a lot more drag on those molecules as they try to move through the gel. The shorter molecules have far less drag, so they can actually migrate further down the gel. And so this friction between the gel particles and the DNA allows us to get a nice spread of DNA. What you're looking at up here is the result. Now, this is actually a gel. What's happened here is somebody has taken a gel like this. They've treated the DNA with a radioactive probe that just attaches to the DNA in the gel, and then they've exposed that gel to an X-ray film. Excuse me. And then they've developed the X-ray film. That way you've got the X-ray film as a record of the gel. But what you see are these little bands of DNA. Okay. And again, if this is the top of the gel here, this would be a long piece of DNA. This is a long piece of DNA. All these tiny fragments down here are the shortest pieces of DNA. One thing I always tell you to remember from Baby Bio, when you see something like this on a gel, this fat band here, and you'll see several fat bands like down here. Here's a fat band. Remember, the width of the band does not indicate how long that piece of DNA is. What does indicate the length of the molecule is how far it migrates. So this is still short pieces, even though this is a fat band down here. This is still a shorter piece of DNA than this one up here, because this one's at the top of the gel and this one is at the bottom. When you see a band that's wider on a gel, what that means is you have several pieces of DNA that are running close together because they're approximately the same size. And so that can cause those wide bands that you see on a gel. All right, now why do this at all? Well, the sequences that each of us have on our DNA molecule are unique to us, and therefore restriction enzymes will cut our DNA fragments, our DNA into different sized fragments. And so the pattern that we see in by looking at gel electrophoresis is like a fingerprint. It's unique. Each of us has a different pattern here, which is why this is also referred to as DNA fingerprinting. And so this is useful for things like determining who a father is in a paternity suit. If you had, say, an unknown soldier and you bring their remains back and you test them and compare their DNA to potential family members, you can identify remains. You can also figure out who might have been present at a crime scene. You can compare samples of blood or bodily fluid or hair at a crime scene to try to place people at a crime scene. So that's all the uses that you can get from this. Now, the other thing that you can do with one of these gels is run what we call a marker, and this particular gel doesn't have a marker, but this would be known lengths of DNA here if this were a marker. And the reason why you would run that at the same time is that if I knew that this was, say, 30,000 base pairs and something ran next to it at 30,000 base pairs, I would know how long this piece of DNA was compared to the marker. If I'm looking for a gene that I would like to use in biotechnology, and I know that the gene that I'm looking for is about 31,000 base pairs, I know that gene's going to run about here on my gel. So when I run the gel, I can then cut out the DNA from the gel. I can actually literally take a razor blade, cut the gel, lift that piece of gel out containing the gene that I want, wash the gene off, and use it for genetic engineering. Speaking of genetic engineering, old school genetic engineering, actually the oldest of old school genetic engineering, is called selective breeding, right? And we talked about that during the evolution unit, where you simply breed things together to get specific traits that you like, and you keep trying to breed those in. The next wave of genetic engineering is what you see in this diagram here. This is recombinant DNA, and this is a review again from Baby Bio. If you wish to create an organism that contains a new genetic profile. For example, in this particular picture, you've got a bacteria, and what we're trying to do is move the human growth hormone gene into this bacterium so that we can get bacteria that create that for us. Then we would use this process. This is how insulin is created. So insulin that diabetics take was first produced using recombinant DNA technology. And I think everybody can agree, even though this is genetic engineering and that might be a little scary, that's obviously a really important use of genetic engineering. Lots and lots of diabetics need that insulin. They need safe insulin. And this technology allows us to mass produce a whole lot of safe, clean insulin for diabetics to take. So, in order to do this, first of all, you take a restriction enzyme like this one, EchoR1, and you pull a chromosome out of the DNA, out of a human cell, and you digest it with EchoR1 to create sticky ends. And here's our little sticky ends, and these are little open hydrogen bonds that you see here. There they are. You also pull a plasmid out of a bacterial cell and you cut it with the same restriction enzyme. And the key here is that, because if I cut it with the same restriction enzyme, I know that the sticky ends that I create here and here are going to match to the sticky ends that I created here and here. Then what we do is we allow these pieces of DNA in a little test tube to hybridize. So we just allow that donor gene, that human growth hormone gene, to attach to the DNA, the plasmid here. And now I've created a recombined or recombinant plasmid. The next thing that I need to do is transform the bacteria. And by that, I mean I need to take this plasmid and I need to stick it back into the bacteria so that now the bacterial cell machinery can start producing the human growth hormone for us. Now that it's got a new genetic profile, it will start to actually transcribe and translate that gene and produce human growth hormone for us. Transforming bacteria in a lab requires what's called competence, and we would have done this lab had COVID not taken over our lives. But what you do as part of a transformation experiment is to go through a heat shocking procedure in the presence of a chemical, calcium chloride, and what that does is basically makes the bacterial cell wall porous enough that it is more likely to pick up our recombined or recombinant plasmid. And that's chemical competence. That's what you're doing. By heat shocking, what you would do is you would actually stick the bacteria with the plasmid. You would incubate it for a period of time on ice, and then you would stick it into a hot water bath for a period of time, and then you would put it back in ice. That's what we mean by heat shocking. So you take it from cold to hot to cold again. So that's how you do genetic engineering, essentially. That's how you would engineer a bacterium. Now, there are a few catches with this. Not all laboratory exercises work, and scientists growing bacteria in a lab can't just look at the bacteria and say, yeah, that one will produce the human growth hormone that we need. So what they will do is usually insert 2 genes for the price of 1 here. And there's a couple ways that you can do this. You can not only insert the human growth hormone gene, but at the same time that you insert this, somewhere over here, you might insert a gene for antibiotic resistance, like something that's resistant to penicillins, like ampicillin. And that way, if you plate your bacteria on a culture dish that contains the antibiotic ampicillin, then the only thing that could grow on that plate would be a bacterium that contained the antibiotic resistant gene. So you know that these bacteria have indeed picked up the plasmid that we're looking for. Another modern way that scientists are doing this is by inserting really cool jellyfish DNA that glows, right, or phosphoresces under UV light. And again, if you insert a gene for that here, then all you have to do is run a UV light over your plates, and any of the glowing colonies of bacteria that you have, also picked up the gene for human growth hormone. So that's a couple ways that you can do that. And there is a really cool, on my original website, there's a link to a SciShow video about why scientists create glow-in-the-dark rats, and it's a description of all of this. Now, that's great for bacteria to create products for us, but that's not all there is to genetic engineering, right? You've heard of CRISPR, right? CRISPR is a technology where you can actually customize the CRISPR complex, which contains enzymes and then bits of DNA. And CRISPR can actually be injected into the body. And instead of going through all of this, what CRISPR will do is go ahead and insert a gene of interest directly into a human chromosome or any other chromosome that you want. That's one way of doing it. There are even gene guns that have been invented to inject specific genes into cells. Ultimately, why engineer a human? Most of the time, what we're trying to do is something called gene therapy, where you could take somebody who has, say, sickle cell disease. We know what that mutation is. If we could inject somehow and splice into their chromosomes the correct form of the gene that's malfunctioning, you could cure sickle cell doing that, right? Which it would be a magical thing. And so that's kind of the reason why some of those technologies were invented. Now, obviously, those technologies could be used for a lot more insidious things too. You could get into a very serious issue with designer babies and things like that. But anyway, that's kind of why they exist. Let's take this back just a step, because the AP exam isn't going to ask you about CRISPR. How do you transform something big, like in this case a plant, or how would you transform an animal, right? In Baby Bio, we talked about inserting the protein for, or the gene for spider silk production into goats, or sheep, I guess it was sheep, so that they could produce spider silk in their milk, and we could harvest spider silk because spider silk is really strong and it's got possible uses in surgery and things like that. And so to do something bigger, you start with this process with recombinant DNA, but now what you do is you transform a bacterium. In this case, you're using an agrobacterium, which causes galls in plants that normally infects plant cells, but you engineer it so that when it infects the plant cell, it would insert a specific gene that we want, like drought resistance or having more vitamin C or whatever you want. And then we would let that bacterium infect the cells in a plant tissue culture. And then what we would do is let those cells grow up, and notice now they contain the gene that we wanted, like our drought resistant gene. As those plant cells grow up, now you end up with a plant that is drought resistant. It contains a new genetic profile. We use bacteria for this. A lot of times in animals, you would use viruses to do this as well. And again, the reason for this is that viruses and bacteria are really good at getting into our cells and messing around with our DNA. And so if we could engineer a virus or a bacteria to do that on our behalf, then we can change the genetic profile of cells. Of course, the viruses and bacteria are engineered so that they're not going to be virulent. They're not going to actually attack the cells and cause damage. But that is a possibility. None of this is foolproof. In animals, you've got an immune system that any virus or bacterium that we engineer would have to get past in order to move the gene into the cell of interest. But it does seem to be work. Presently, I think there are several trials for various gene therapies using this transformation. One is a nasal spray that contains a virus with the correct form of the gene that causes cystic fibrosis. I've heard of another trial in which they've injected people with a bacterium, I think, or a virus that injects the gene that corrects sickle cell disease. So it is something that's being worked on. Another technology is polymerase chain reaction, or PCR. And this is not really doing any kind of manipulation at all. Um, so this is not genetic engineering that you're looking at here. One of the big problems for scientists is oftentimes if you're at a crime scene or if you find a mummy or something like that and you want to get DNA out of it, you might only be able to get a very, very tiny sample of DNA. And what PCR can do is magnify a sample of DNA. So within hours rather than days, a machine and a specialized enzyme called Taq polymerase can actually create for us hundreds or thousands of copies of DNA in a very short period of time. Um, the machine that actually does this is called a thermocycler. And basically, you need heat to denature or unzip the DNA molecule, but then you need to cool the DNA molecule down once um, the new nucleotides have been added through replication. Um, and that promotes what we call annealing, which is the hydrogen bonding that causes the two strands to come together. And so there's a machine that goes through these thermal cycles to do this. And essentially you have, and like I said, this is going to require a Taq polymerase, a very specific kind of heat stable DNA polymerase to do this. Um, so they got this from a bacteria that lives in really extreme heat conditions is how it was isolated. But essentially what you do is you start with a solution containing the template DNA strand and you have a bunch of primers in it. And these primers are going to attach to the region of the DNA that we want to amplify. You're going to then heat the DNA up to cause it to denature. And then the primers are going to anneal to the actual DNA molecules. Taq polymerase is going to create the new strands of DNA. And then it's going to repeat the cycle again. And you're going to go through another denaturing period, replication period, and then annealing period. And it just keeps going through this. You repeat this cycle 20 to 30 times, and you get a whole bunch of copies of DNA. And then, like I said, you can do whatever you want with that sample. You can use it for gel electrophoresis to do some identifying, all kinds of things like that. Some other technologies that pop up here. One thing is to talk about the Human Genome Project. And the Human Genome Project was a project that was set out, it was a multinational project to sequence and map every gene on the human chromosome. So there was a three-step approach. The first thing is they created linkage maps, which we would have done in the Sordaria part of your mitosis-meiosis lab if we had been together. But essentially you're using the percent of recombinants to determine how close together two linked genes are on a chromosome. Then they use that to actually construct a physical map, and then once they had kind of where all of the genes were on the chromosome, then they actually sequenced every single gene. And this was a massive undertaking, and it gave us a lot of insight into how genes in the human body work, and how chromosomes are packaged and things like that. Ultimately, it's this kind of technology that led us to do the kinds of things like gene therapy and stuff like that. And so it was a really important project. Also, what comes up with biotechnology is stem cells a lot, and we've kind of talked about these before. Stem cells are used in biotech for a number of reasons. One of the reasons, obviously, is to test sometimes new therapies, test drugs and things like that on stem cells. So rather than, if you need to see how a drug is going to affect a human being, rather than giving the drug to the human being, you could give it to human stem cells first. And see if it affects those cells. Then you're not hurting a human being in a drug trial. You're just hurting a collection of cells in a lab. Scientists study stem cells and how they develop, and they look at the the way that genes are turned on and off in cells from stem cells from people who have illnesses like diabetes to kind of see what are the steps that cells go through to become cells of a diabetic person or something like that. So they're used as well. Their use is controversial because to get the best kind of stem cells, which are the embryonic stem cell lines, obviously you have to destroy an embryo. Although that is not the only way anymore. You can actually, scientists have figured out a way to take specialized cells and revert them back to an earlier state, to an embryonic type state where they can actually be used. Also, umbilical cord blood contains a lot of these embryonic stem cells. So if you harvest that umbilical cord, you can get stem cells there too. So using stem cells has become a bit less controversial now that there are some new technologies to make to be able to get stem cells a little easier without hurting embryos. All right, cloning is another hot button issue. Now, cloning does not involve any kind of genetic engineering. You are making a copy. In this case, we're talking about, by the way, we're talking about what we call reproductive cloning. The word cloning really means to make a copy. So technically, if your cells are doing mitosis, they're cloning, right? Identical twins are clones of one another. Those are natural forms of cloning. But when most of the time people talk about cloning in biotechnology, they're talking about reproductive cloning. And you can obviously plants are you can do really easily because plant cells retain a whole lot of stem cells within all of their tissues. So you can actually propagate plants where you just take a cutting of the plant and stick it in some soil or some water with some hormones in it, and those stem cells will take over, and some of the tissues will redifferentiate even. So they can actually de-differentiate. Cells at the tip of that cutting can de-differentiate, and then they were cells of the stem, and they can redifferentiate to become cells of the root. So it's really easy to clone a plant. Cloning an animal, obviously, is a bit more difficult. And the most famous clone, the first cloning experiment that was successful, was Dolly the sheep. And I have a picture here of how Dolly was created. You took cells, and by the way, these are differentiated cells that were taken in this process. And they took these cells and de-differentiated them a little bit, these mammary cells, and then they pulled out an egg cell from a donor sheep, and they took the nucleus out of it. They removed it, and then they used electric shock to take these mammary cells that they de-differentiated and fuse them with the egg cell, so that now this particular egg has a brand new nucleus inside of it. And because it's an egg cell, it's going to do what egg cells do, fertilized egg cells do, because it's got a diploid nucleus in it now because it came from an original cell that was differentiated. And it's going to start to divide and form an embryo. So that embryo was put into a third sheep, a surrogate mother sheep, and allowed to gestate. And then, after the gestation period, Dolly was born. And Dolly then is genetically identical to this sheep here, to the donor of the mammary cells, because it's that cell's nucleus that Dolly has. So that's how it happened. It was done by a Scottish scientist named Ian Wilmot in the 90s. But there are some issues with organismal cloning. It's inefficient. Only a small percent of cloned embryos can develop normally. And a lot of times there are defects. Dolly herself had to be euthanized at a fairly young age because she suffered from a lung disease that would normally affect a much older sheep. Scientists think this has a lot to do with maybe some epigenetic factors of these mammary cells that were used. That possibly the DNA of those cells didn't act like embryonic DNA because it was coming from a differentiated cell. So they're not really sure. It may be also that this DNA had already accumulated a whole bunch of mutations which would mean that even though this is a baby sheep, it had old DNA with many more mutations in it, which would explain why Dolly got a geriatric disease at age 6. So scientists have been working on this. This is, I mean, in the scheme of things, it's actually not a new technology since it's been around since the 90s. And so some improvements have been made since this was first developed. All right, finally, just to kind of summarize, there are a whole bunch of reasons why we do biotechnology, right? There are some medical applications, right? We can help to diagnose diseases. We can develop pharmaceuticals. We can develop foods that contain vaccines, for example, or foods that are more vitamin rich, or something like that. So there are obviously some agricultural apps to that as well, but they kind of border on agriculture and medicine. Also, gene therapy, right? Fixing bad copies of genes. Forensic evidence we've touched on. We can also engineer organisms to clean up the environment for us. So there are some bacteria, for example, some plants that have natural ability to remediate, like things like crude oil. Or toxins in the environment. If we could take the genes that make those organisms capable of doing that and implant them into other organisms, then we can engineer organisms that are actually able to clean up our environment for us. So there's a whole bunch of reasons to do biotechnology. This is a process, though, that needs to be done very carefully. There are a lot of safety questions about genetically modified organisms or genetically engineered individuals. Those are called GMOs, by the way. There are problems with technologies. There are a lot of issues, for example, of genetic engineered organisms being released into the environment and what impact are they going to have on the environment. Obviously, issues with obtaining stem cells. Creating organisms with medical issues since the clones don't seem to be as vigorous as the real thing. CRISPR in and of itself, if you followed the news, kind of the newest technology, has a whole lot of issues with it. There's a lot of excitement that surrounds that technology, but also a lot of fear about what would happen if somebody did the wrong thing with it. So the key here is we as a society have a long way to go with where to draw the line with research. And I'm mentioning it here. This is not a short discussion to have, but it is something that's in the forefront of everyone's minds.